I'd like to begin with just a few basic introductions, if you don't mind. And Trevor, as a newcomer, really, can we begin with you?
Sure. So my name is Trevor Smith. I'm the Senior Marketing Manager for Cell and Gene Therapy in North America for Terumo Blood and Cell Technologies. My experience in cryopreservation at Terumo BCT has really come from our Finia fill and finish system. I've also had experience as the product manager at BioIVT for our Leukopak and immune cell portfolio. And I've personally done my fair share of freezing and thawing cells during my time at Cytiva doing R&D.
Thanks, Trevor. Priya?
Hi, Rob. I'm the Chief Business Officer at OrganaBio, and I also lead our process development and manufacturing and serve as our laboratory director. So I really sit at the intersection of the science and the business, marrying those two to move the organization forward. I'm a founding member of OrganaBio. Before this, I was a founding member of Rooster Bio, and OrganaBio is actually my third startup that I've been involved in. So either an adrenaline junkie or a glutton for punishment, depending on who you speak to, right, Stella? So I'm a PhD in biomedical engineering, spent many years at the bench with stem cells in the context of tissue engineering, cell therapy, and regenerative medicine more broadly. And so I've cryopreserved a lot of cells as well over time, different types of stem cells, different types of immune cells now. And as we are a services provider and are providing tissues and cells to cell therapy developers at OrganaBio, this is a constant conversation that we have around the needs, the limitations, the advantages of cryopreservation.
Thanks, Priya. And Stella?
Well, hello, everyone. I'm Stella Nook. I'm the CEO of Licarda. I spent over 25 years in this wonderful industry, starting out from the bench and really transitioning from science to business. So similar to my colleagues here, I've spent a lot of time passionately advancing our therapeutic treatments. I spent a lot of time focusing on cardiovascular and oncology areas. And with regard to cryopreservation, I think that 20 years ago, we weren't thinking about it at all. 10 years ago, we sort of did with what we had. And now it's at a critical tipping point of, well, we need better solutions. Hence, my pedigree normally goes into large pharma, biotech, perhaps CDMO space. And I jumped on Licarda as a drug delivery company because we need different solutions for advancing cryopreservation. So I'm thrilled to be part of this talk.
Thanks, Stella. I'll stick with you just now because you mentioned that the industry is at a tipping point. What has brought the industry to that tipping point? And how's the demand for cryo changed with the development of advanced therapies?
Well, great question, because actually I was thinking about it myself preparing for this interview, and I was trying to think, what was I doing 10 years ago? And I think 10 years ago, the demand for cryopreservation was really driven by sort of storage of biological samples, right? We were storing biological samples in research in universities, maybe reproductive medicine. And then we had the rise of advanced therapies and the cellular and gene therapies. The demand has significantly increased. So I identified that to myself as well. That was the critical point for whatever the minimal we needed to do, we now need to expand on that. So for instance, the global market for cryopreservation equipment, that's what I was able to quote, was around 2.5 billion in 2014, and now projected to reach over 8 billion by 2025, driven by expansion in cell therapies and regenerative medicine. So what we had before is utilization of primarily DMSO. We're also learning more about the DMSO and maybe there's new players on the market with non-DMSO, and we now realize that, oh, well, we can't use DMSO in these conditions like cardiovascular, glioblastoma, and other therapies. So that as we evolve in our ability to advance cell therapy or other modalities, we realize what we can and cannot and should use. And then to continue in that, we also realize the cost to the system as more of these drugs launch. We also realize the cost of cell therapies per patient. And we spend so much time and passion launching these products. We want them to be available and accessible. And yes, we can scale up manufacturing, but then the cost of transportation and logistics for these products are just skyrocketing. So we need to start solving this probably earlier before sort of it hits that budget line and we realize that we're at the mercy of, well, the price is what it is for liquid nitrogen transportation. So complex story, but that's how I was thinking, well, we're at the tipping point of needing to solve this.
Priya, do you agree? Can you elaborate in your own words?
Yeah, absolutely. I agree with everything that Stella said. I think as we see more and more cell and gene therapies emerge into the industry, one thing that I think we're all very passionate about, as Stella mentioned, is accessibility of these therapies and democratization of access of these therapies globally. So while we still see a strong preference amongst many cell therapy developers, especially I'd say the more nascent biotechs, for fresh materials because of the inclinate or the fact that they are closer to the native tissue or the native cells and they haven't been manipulated as much. Fresh tissues, fresh cells pose a real logistics challenge, not only when it comes to the time of manufacturing and the storage or the transportation and storage of these materials. You're really running up against a clock. And so cryopreservation allows for extended storage and it allows for transport globally and deployment of these therapies globally where you're freezing these cells or these tissues in time. And so moving to a cryopreserved product is something that large pharma has already embraced. A lot of emergent biotechs, more established biotechs have embraced and continue to move in that direction. And I think until we have better processes for maybe lyophilization or more next generation solutions to circumvent cryopreservation, it's going to continue to be the path forward for deployment of these therapies. Also, I think the gold standard moving forward someday might be the cryopreservation of entire organs, right, and being able to transplant those organs. And so that's something that I think the field continues to iterate on and move towards, not just cells, but entire organs and tissues for transplantation to solve a lot of the issues that we have in the transplant realm and the availability of issues for the many people waiting organ transplants and who maybe do not have access to cell and gene therapies today. And as we see cell and gene therapies continue to expand in terms of their indications beyond cancer now to autoimmune disease, and then beyond that, we're going to continue to see the need for these technologies. I think Stella's right. Cost is definitely a major consideration here, along with the regulatory hurdles that might exist. And so the industry as a whole needs more standardization in this realm. We also talk a lot now about decentralized manufacturing. Cryopreservation does offer some advantages, again, in centralized manufacturing and standardization on that front. But as we move towards decentralized manufacturing, point of care deployment of these therapies, standardization and cryopreservation, thawing of these therapies, and the regulations globally around this are going to be very important for the industry to move forward.
Thanks, Priya. Trevor, how is the demand affecting your business, your line of work? What are your thoughts?
Yep. So I'd have to agree with my colleagues that there's an increase in demand for these cryopreserved products as we learn more about these treatments and these therapies. I think operationally, these treatments are themselves the very definition of personalized medicine. So, while it's table stakes in the operational process to collect and ship patient material and do the manufacturing into the final product and ship that back for reinfusion, it's really no small feat to do all of that. And I think, logistically it's challenging, aligning these different schedules for these different sites, be it in centralized and decentralized manufacturing. But cryopreservation gives you a path forward to make it so these things that have a lot at stake get there in time, with the quality that's needed for the patient.
Trev, can you just elaborate for me, please, the advantages of cryopreservation, storage and transportation wise over conventional methods?
Yeah, for sure. I mean, conventional methods, I view as keeping the materials fresh or even chilled. Again, to keep it as close to, the native cell and tissue populations as possible. While those forego the risks of viability that are today inherent to a normal freeze-thaw cycle for cells, cryopreservation in effect stops the clock on therapy manufacturing. So this in turn eases the burden of scheduling at the collection site from the patient to better align with the manufacturing suite availability downstream for more seamless transitions into the manufacturing process. I would say cryopreservation also enables different dosing strategies, such as multi-dosing for patients, which really gives more flexibility for the advanced therapy developers to, create a product that's more beneficial to patients.
Stella?
Well, I completely agree. I would even explain it further to say that, look, even with cryopreservation a decade ago, what we knew before was, how do you test if the item was cryopreserved? We had very limited tests, basically comprised of cell viability and functionality. And now that cryopreservation is becoming standardized, right? We expect to have some form of it, and we look to optimize it. The cryopreservation, for example, with CSS platform that we offer, can go a lot further. We can include cell survival rates, reduce cellular damage. We can extend storage duration without compromising efficacy, and we can test not just for viability, we can test every step of the way how cells are functioning in that environment. So having a precise and reliable preservation methods are critical for us to assure that the therapies that patients get will have maximal therapeutic benefit.
I notice you're not in agreement there. Anything to add of the advantages?
Yeah, we've talked about the reduced time pressure, the deployment, the ability to, interrogate these cells more routinely and in a more meaningful way, and standardization. But one last thing I would add to that is also perhaps reduced risk of contamination, right, with cryopreserved products versus fresh products, fresh tissues. So that is another added benefit of cryopreservation when you talk about advanced therapies and the safety of these therapies, the integrity of these therapies. Also, biobanking, and as Stella mentioned, the IVF field is a major driver of cryopreservation in healthcare and medicine. But when we think beyond fertility and that field, biobanking is still something that we do routinely, of tissues, of cells, of cord blood units, what have you. And we have newer options coming to market now, and ideas about first-line treatments for patients versus, second, third, fourth, fifth-line treatment, right? Today's immunotherapies end up being after chemotherapy, after radiation, after some sort of monoclonal antibody or any combination thereof. And many of these patients' bodies are ravaged by the time that we are able to go in and leukapheresis them, for instance, in the context of an autologous CAR T therapy. And so we're seeing the thought now about more prophylactic apheresis of these patients, maybe as soon as they receive a diagnosis, and banking those materials so that then when insurance companies and regulators and everyone says, okay, nothing else has worked, now we're willing to try the CAR T therapy, that material is available from the patient for their use back from when they were a lot healthier and before their bodies were ravaged by all these other therapies. The hope is certainly that these therapies get pulled up to being more first-line therapies rather than last hope and last dish chances. But in the meantime, cryopreservation also allows for biobanking, which is very important.
Okay. So the advantages and the benefits of it are pretty clear and self-explanatory, I suppose. Are there any inhibiting factors that advanced therapy developers might face in accessing or utilizing cryo-based solutions? Priya, can we continue with you, please?
Sure. So Stella already talked about the technology advancements that are needed and the cost therein, right? So you do want to bring, I think, automation to bear on these technologies for scalability and sustainability, and eventually for driving down the cost. And also standardization. But the cost of some of these technologies or capital equipment that's needed for cryopreservation can be quite costly. And then we look at how we scale that, right? Do I need 100 CRFs? Do I need 100 of whatever? Or is there a scalable solution with this platform that will allow for large scale manufacturing? So I think equipment is definitely one area that is a focus for the industry and will continue to be so, and is a current limitation. There's also, workforce development that needs to be done. Cryobiology is a very specific field of biology, cell biology, and there aren't a lot of cryobiologists out there with the very distinct knowledge of what it takes to cryopreserve cells, tissues, living materials, and for the vast variety that we need. Again, if we are to, an NK cell responds very differently to cryopreservation than an HSC, and hematopoietic stem cell, versus just a mesenchymal stem cell or a fibroblast. And so you need someone who understands cell biology, who understands the physics of freezing and temperature changes and nucleation. And, it's really a very multidisciplinary field that not a lot of people have a lot of experience and knowledge in. And so we need more technical experts in the field to drive it forward as well. And then finally, the regulatory hurdles, right? DMSO has been quite ubiquitous and is used routinely. We have to get our regulators comfortable with the newer technologies that are coming to market, the newer solutions, and find ways to make sure that all the validations and the safety profiles, everything is behind it, to allow use of these new reagents and materials.
Trevor, any inhibitions?
Yeah, for sure. Well, from my perspective, obviously, Terumo Blood and Cell Technologies providing a device specific to this area. I think the biggest challenge for adoption of automation is simply that there are so many other areas to focus on during therapeutic development that too often cryopreservation, the final fill and finish step, is often deemed a lower priority when compared to bigger steps like expansion or transformation that really forms the crux of the therapy itself. So we've even reached out to developers, and we hear this strong desire for them to automate and standardize their processes. But the timeline for adoptions are often later during their clinical trial process development, simply because of these other areas and the manufacturing process that kind of get more attention paid to it. I think Priya also made a good point about the access to expertise in the field. Certainly, the folks in charge of cryopreservation after a manufacturing process are going to be a different talent pool than those doing cryopreservation immediately after collection. And how do we train and make it so that both of these sites and both of these groups of people have the same level of expertise, so that you get that consistency even within the same process of cryopreserving starting materials and cryopreserving the final product such that it's the same.
Thanks, Trevor. Stella?
Well, my colleagues are absolutely spot on. We are just amazing. We are very much aligned and very passionate about this field. So in my mind, there are really 4 factors that are critical for us to solve. So my colleagues already talked about, so the complexity of logistics, cost, the infrastructure requirements, the need for equipment, the temperature control environments, the backup systems, the cryogenic conditions. The infrastructure is expensive, so that was already said. Ray also mentioned about regulatory and safety concerns, right? So handling liquid nitrogen possesses risk. DMSO possesses risk. So we need to do better as an industry to educate ourselves, our colleagues, FDA about what we're doing now and where we need to go. And there needs to be more educators. So again, it was very well stated. And then number 3 would be, how do you make it sustainable? I don't think we can accommodate the rural environment and, creating the single standing units next to each hospital or oncology center. So how do we create sustainable solution and not overutilize energy consumption, which leads me to number 4. I think that we haven't talked about the environmental impact of production, transportation, utilization of liquid nitrogen,